right? So this is the general one, okay, and very typical for all chemical, physical, biosensor. Okay, these are the things that has to be done. Right? So these are definitions. Okay, I will of course share with you the actual activities, yeah, lab activities or research activities that has to be carried out in order to do all this testing, characterization for this biosensor or chemical sensor. Okay, so let's look at the first one. Usually we start with response time. Once we are clear on the principle of how it works, okay, meaning that we are clear in terms of interaction between the analytes and the bioreceptors or chemical receptors, means that part is,
Rehat kejap semayang.
is clear, means that you have to do that at the early stage. You have to confirm the interaction is very selective, good enough, good to go for the development of sensors, chemical sensors or biosensors. Then we start with response time. So response time, it is based on the signal generated by the transducer. So remember the interaction between the analytes and the receptors will be shown, will be transduced, will be giving out, the signal will be given out by the transducers. So based on the transducer's signal generated, we can measure the response time. Whether the response given by the transducers is up to whether it's electrochemistry or whether it is optical. We need to see the generated signal. The response time has to be relatively fast. Because if we're talking about 24 hours, even 2 hours is considered very long for sensors. Because the purpose of having chemical sensors, physical sensors, or biosensors is to have a portable and a very fast response, okay? Very fast response sensors, okay? So if we have to do analysis like in the lab, step by step, and requires you to have like, probably the results can be obtained in 2 days or 24 hours, then it defeats the purpose. It's just not, can be defined as sensors, okay? So the time necessary to come to an equilibrium means that at least the response time has to be very fast, but showing the correct response. What does it mean by equilibrium? That it does not change. Yeah. Once we identify that the signal generated should be, for example, electrochemistry, we should know the potential should be around, let's say, -1, and the current should be around 20 microamp, for example. Okay, and then we know that will be the most stable signal that should come out from the interaction. So let's say if the signal fluctuates, sometimes it goes up, sometimes it goes down, then it is not considered a very good to be developed into further into sensors. So response time is very important to be identified at the first stage of development, at the first stage of characterization. Okay. Once we have a good response time, we can try to look at the precision and accuracy by doing repeatability or reproducibility. What is repeatability? We do the replicates. Yeah. So we look at the precision and we look at the accuracy. Accuracy, usually we don't do at the beginning. We do during the validation time, which is not shown here. Okay, usually we delay that. We focus on the precision first. Why? If like we repeat the replicates or the measurement with the same set of parameters, with the same set of conditions, we should be able to get the same result. As I have given, the one that I give example just now, if the interaction should show potential at -1, and the current should be around 20 microamp. So if you repeat the same protocol, if you repeat the same material under the same condition, then it should be giving more or less plus minus 5% of, of standard deviation, yeah, error for that particular measurement, okay? So that is very important. We call it repeatability. Every time we do it with the same sets, then it should give you the same results, okay? So if you cannot get that, then we have to change. It's either we change the transducer or we need to change some parameters using the transducer, or we change the receptors maybe. Okay, we need to change the receptors. So those things are very important to be looked at. And then next one, okay. So selectivity by right. At the beginning, I told you that we should evaluate that at the beginning. Meaning that if you're working with a DNA probe, if you are working with antibody, or if you are working with chemical reagents claimed to be sensitive or specific towards that particular analyte, it has to be tested first. Okay, it has to be tested by means that's suitable to check on the binding. Okay, sometimes we do UV visible. Sometimes we use fluorescence. Sometimes we use different methods like titration. Okay, we have to test. But there are still some selectivity that we have to do towards the end, okay, of the characterization of sensors. So now response time done, repeatability, focus on precision done. We have to do stability study. Okay, stability study usually stability and lifetime usually connected. We can do once, like we skip the sensor set for 1 month, or from studying with probably weeks, month, and we look at the performance, whether the performance remain the same or it degrades with time. Okay, so why we do this? Because if we are to commercialize, then we have to tell the lifetime of the sensors, okay, expired by, okay. So stability and working lifetime is connected, so you can do together. And reproducibility. Okay, what does it mean by reproducibility? Sometimes the sensors are a disposable basis. What does it mean? If you do it one time, just like your the strips, sometimes the electrode also, it is good for only one measurement, and then we have to dispose. So it is disposable basis. But sometimes we can also regenerate. We can use the sensor back just by probably washing and putting in some chemicals or some biochemicals. It can be done, depends on how you design the sensor. So reproducibility is measuring the quality of the sensor once we do the regeneration. So if we are able to regenerate the sensor, and we have to check on whether we will get the same response, the same signal after being used a few times. So depends on how many that we can use and what is the degradation of the sensor once we regenerate, right? So this one, sometimes, as I said, some prefer just disposable basis, so you don't have to do the reproducibility, and some to be able to reduce the cost, we can design also uh, regenerable sensor. Okay, right. We have done this. Okay, precision, reducibility. And now the next one, which is very important, is sensitivity. That depends on, yeah, if you are doing something quantitative sensors, of course, sensitivity will be very important. But if we are doing like yes or no, like the strips that you use for COVID, it's like yes or no. But still, we need to know about sensitivity, whether it can give you results, okay, or it does not give you any signal. So sensitivity is not just about quantitative, it also applies to qualitative sensors. If it is not giving response, then it's no use to be developed further for sensors. So how do we test the sensitivity? Depends on the method, the transducer. So if it is quantitative, usually we use calibration curve, okay? Just like what we have learned in statistic. Okay? You have to study the linear range, what is the concentration of the analytes that give a linear range until what limit, okay? Because we understand that there is always limitation. So let's say if you're using absorbance, okay, in optical sensors, you're using absorbance parameter, then there is Beer's law, that meaning that the linear part only uh, uh, can be obtained for certain concentration. So after beyond that concentration, then you will have a fluctuation maybe, or you will have a nonlinear uh, plot, which is not suitable for uh, quantitative application of the sensor. So working linear range and sensitivity comes together, all right? When we do the concentration analysis, uh, concentration effect on the signal, then we can know the working linear range. And in the end, we can calculate the limit of detection or limit of quantification so that we know whether the sensors is good. Okay, so for example, if you are to detect the toxic level of plumbum in river water, so it has to be very good enough to detect down to certain part per billion. So if your sensor is not good enough to detect down to part per billion, then it's not suitable to be developed further. In terms of application, it's not very practical. So we have to know the sensitivity. Okay, now let's say if we have done all that, response times are good, the repeatability are good, and then the stability is acceptable. We start to check again selectivity. So I remember that I told you selectivity, we evaluate first before we proceed with all this. But at the end, we also run another selectivity study by introducing interferences. What does it mean is, let's say we are doing a glucose sensor and we are detecting inside serum, then we have to subject the sensor towards other compounds or other things that exist in that serum. Okay, let's say there are proteins, there are urea, there all sorts of things that can exist in the serum. So we have to test the sensor that we develop with this kind of compounds that exist in the serum. So let's say we have another issue like, one of the best is water, right? So if we are testing wastewater from certain industry, then we have to know what are the existing chemicals or what are the possible other compounds that exist in that water, commonly exist in that wastewater. We know already, so we will subject or we will test the sensors towards those compound, towards those chemicals, and look whether it gives a very significant interference um, signal towards the uh, actual analyte that we wish to uh, detect. So this is very, very difficult. This is very difficult. Even though even though we are working with supposed to be very selective, like DNA, antibody, and other proteins, okay, they do interfere. Apart from, yeah, even though we have tried to exclude, like having a very good, a very selective chemical reagents or biochemical reagents, this selectivity issue is still a big, uh, a problematic to anyone who are developing sensors. Okay, so these are typical. These are typicals that you do, right? But before that, yeah, because since sensors is about manipulating the surface, yeah, we have to work with like electrode surface. If we are talking about optical, then we have to talk about, we have to use also some uh, that we need to embed on the surface of the sensor. So this characterization also requires you to do all this surface analysis. Okay, like this one, it is called EFM. This is, I forgot, this is contact angle. Okay, this is contact angle. This is AFM. And sometimes we do FTIR to check on the surface. Okay, we have to do this also apart from this one. Okay, so in order to develop a good sensor, we have to do surface analysis, including this among contact angle, FTIR, AFM, yeah, atomic force microscopy. So these are things that we need to do before we can further develop. This is example of atomic force microscopy. Okay, and we also need to, if we are incorporating certain nanomaterials or metal oxides like quantum dots or carbon dots. Okay, carbon dots usually we do FTIR. Quantum dots we do XRD. Okay, to confirm on the material that we have developed, to confirm on the modification on the surface of the sensor. So we have to do morphological study like using SAM. So if you are using a nanomaterial, then we have to confirm using TAM. Okay, and this one I'm not very familiar, scanning tunneling microscopy. And if we are working with like metal oxides, yeah, we have to confirm the elemental analysis by using XRD and all this. Yeah, XPS will confirm certain things like nitrogen. If we remember about DNA attachment, um, antibody attachment on the surface of the sensor. So this will help us to confirm there is attachment, there is immobilization of chemical or biochemical on top of the sensors. Yeah, all right. So these are some of the methods that you can use if you are working with nanomaterials. And this is what you wish to know. For example, If it is about oxidation state, then you can use XPS, elemental analysis composition, XRD, ICP. So concentration, definitely, if you are working with gold nanoparticle, you have to use UVVs, yeah, to identify the concentration. If it is size, temp, shape, also temp, surface charge, okay, zeta potential. If we need to know the size distribution, so this kind of optical properties, magnetic properties is usually uh, not that, but so if you are working with magnetic nanoparticles, then you have to do the magnetic properties. So this has to be done before this characterization. Okay. So this has to be confirmed on the surface and this one on the material if we are using some types of material. Okay. Right. So let me open the other one, the first one, which is on the electrochemical. So share this one. So what I'm going to show you is more on examples, yeah? Okay. Just a moment. Okay, you can see the slides, yeah? But it's in PDF form. So the slide is on electrochemical after sensor with dual bioreceptors. Can you see that? Hello? Anyone?
Yeah, we can see.
Yes,
All right.
yes.
Yeah, I need to confirm because when we change. Right, so what I'm going to show you is development of biosensors for detection of tuberculosis. Okay, so you can see that what I have taught before, what I have discussed before, it should be some of it integrate in this development of TB biosensors. So in this case, we are talking about electrochemical technique as the transducer and the usage of aptamer. Okay, the usage of aptamer to detect tuberculosis bacteria. Okay, so of course we start with introduction and all that, right? Let's just go through this. Okay, so very important in development of sensors, chemical or biosensors, is to understand the principle, whether the principle is, well, when we haven't done the work, we have to like confirm through literature. So in this case, uh, the hypothesis is we are using two kind of biomarkers, okay, MPT64 and CFP10. So these two proteins are secreted by the bacteria, tuberculosis, Mycobacterium tuberculosis, the bacteria that cause the TB detection, TB infection. And it is from the literature that this MPT64 and CFP10 protein is secreted in the whatever it is infecting, whether it's lung or in other parts in the body. Okay, so this is hypothesis, and based on the aptamer that we studied, the aptamer is very selective towards MPT64 and CFP10. And from there, we can have two interaction. One is with MPT64, MPT64. So we put the aptamer here for MPT64, and we put another aptamer for CFP10. So the transducer here is electrochemical, right? And the principle is that once the interaction between the aptamer happen with the CFP10 and MPT64, we can observe a signal generated from the electrochemical device. Okay, so this one, okay, literature review. So of course, when we do research, we have to confirm at the beginning just from literature, okay, to confirm on what we hypothesize. Okay, right. So this is where the important things happen. You need to incorporate the typical characterization of the sensor. So these words, like what I have discussed just now, the sensitivity, the specificity, reproducibility, stability has to be checked, has to be studied. But before that, because in this project, the electrodes, the electrode used was embedded with a salt called diazonium. Why we need to embed this one? Because according to literature, it will help enhance the conductivity, okay, because it's salt. And the other one is to help us to get the functional groups to attach the aptamer and the antibody set. Okay, so now we have to study the surface first. So you can see that from the discussion just now, I mentioned about contact angle, XPS, FTIR. So this one are things that we do before we proceed to the sensor characterization. Okay, so we do the surface study first. And also, since the aptamer also is the bioreceptors, so we need to test the binding capacity with the biomarker, which is the CFP10 and MPT64. So you can see that before we decide to further develop into typical characterization of sensors, we have to do the binding study, whether it does give a very selective, okay, and then we have to do sensor characterization, surface characterization. And once we confirm this, it is good to go, then we do this sensor characterization. Okay, so the one that I didn't mention just now is the accuracy. Remember, I didn't mention about the accuracy. So accuracy is usually what we call as validation. So what does it mean by validation? Since we are doing TB, tuberculosis, we have to validate whatever sensors that we are doing with standard technique. So in this case for TB, the standard technique is microscopy and culture-based. So means that the same sample from patient is subject to our sensors, and at the same time is run by the hospital using microscopy and culture. So we need to compare. We need to do the correlation study. So remember the word correlation from statistic. Okay, we do correlation study between this sensor and the standard method. Okay, right. Okay, this is how it looks. Okay, not necessarily everything will be the same. So we do aptamer binding study and then look at the performance, whether the aptamer, which is the biomarker or the bioreceptors, do have a good binding towards the biomarker. Okay, both MPT64 and CFP10. Okay, so I'm not going to discuss about the results, but these are the steps that we do. And then next is the surface modification, whether it give what we want, whether the diazonium is attached onto the surface of this sensor. Okay, this is what makes a difference between the transducers. So in this case, we're talking about electrochemical transducers. So we need to do electrochemical characterization for the electrode. Okay, so it is different when we do optical. It does not require this. Okay, since we do the isonium just now, so we look at the FTIR and then XPS, also contact angle to confirm the diazonium is there already. Okay, to confirm. And then we have to do some what do you call conditioning. Okay, so conditioning here we have to confirm about the electrolytes, supporting electrolytes, buffer. Okay, that one depends on what kind of sensors that you are developing. So some conditions like buffer, electrolytes. Okay, so for electrochemical, we have to check the applied potential, the concentration of the supporting electrolytes. So it has to be done. And then we have to do the concentration analysis of the bioreceptors to make sure which concentration is the best. It is not necessarily the highly concentrated is better. Sometimes it sort of reduce the signal. Okay, so we have to check all this incubation time. Okay, there's a lot of things to do. And then, okay, this is where we start to do the sensitivity wise. Okay, so you now look at the LOD. We don't do LOQ, we do LOD only. Okay. Now you see the word selectivity, reproducibility, where we tested whether it's regenerable or it's not generable, stability, and the last one. Once all is confirmed, we do the real sample analysis, means the sensor is now subjected to the patient's sputum sample. Okay. So once we have done that, we do, okay, sample validation, comparing with smear microscopy and TB culture, right? Yeah. Okay, since there are certain parts, but these are certain characterization. Okay, so for electrochemical analysis, for electrochemical transducer, we can do a lot more apart from just now you can see that they study the electrolytes, and they also study on applied potential, but there are other things that you can do for electrochemical analysis, for electrochemical based biosensor. We do like scan rate study, and then we do effect of pH also. So that depends on the situation, whether you need to do that or you don't have to do that. Okay, one last one. Okay, if you don't have any question, I'll stop share this one. I'm going to share optical. Okay, example of optical. All right, this is going to be a bit short. Sorry. Okay, now you are seeing science fund monitoring, correct? Everyone?
Yes, doctor.
Right, okay. So in this case, we are developing also for tuberculosis, but in this case is optical based. Okay, and to be specific, we are talking about color change. Yeah, that's why it is naked eye. Well, the name is plasmonic ELISA, so anyone is familiar with ELISA. So I'm not going to discuss the detail of the principle, but it is very important. In terms of hypothesis, we confirm, understand the interaction first. Okay, so what is this all about? Is once the interaction happen, the positive sample, okay, will be blue color, whereas for the negative sample, it is red color. So how does it comes blue or red? Um, it's quite difficult for me to explain, okay, but we have confirmed that. Okay, this is another additional explanation on the principle, okay, which is very important. At first stage, to confirm, to do some lab to confirm just the principle is correct. Okay, so you can see here the blue color and this is the blue color and the red color. So blue confirm positive and red is negative. Negative means there is no tuberculosis mycobacterium in the sample. All right. So the first thing, as just like the electrochemical, in optical also we have to identify the interaction first. Okay, all sorts of chemicals involved. Okay, you can see whether the formulation, the ratio, because you can imagine there are so many things involved here. We have antibody, a few sets of antibody, and we have enzymes involved. So when we work with a lot of biochemical, then everything has to be in the right portion. So all these things are not just come out from thin air. Everything has to be optimized. Everything has to be studied until we come up with these 4 microliters, 5 milligrams. So this is the final version. So we start from scratch, understand how much in order to get the good color. So things have to be done first. All this, yeah, and then at the same time, in this case, we are also involving like a gold nanoparticle. That is the main one. And it can swap from gold nanoparticle to gold clustered. That's what you get, the blue color. So all these are actually very challenging for us. And then we start to study on how to embed the antibody onto the ELISA plate. Yeah, this is ELISA plate. Okay. Right. So once we do that, we are able to build the antibody stacking and then look at the reaction. So there are other things that we have to do since it is not uh, listed in the typical characterization. But one thing you might notice, response time, yeah. We also have to optimize all the chemicals or biochemicals involved. So look at the concentration of antigen, concentration of antibodies, and if there is any other chemicals involved, so we have to study the optimized concentration of the chemicals. How do we dilute the enzyme? How much we need to dilute? So all this has to be done first, and then just like now, we need to study the nano materials, then we do confirm the nano size of the materials, and then start the color. And then we start the response time. So this is repetition, repeatability in terms of precision, whether we will get the same color every time, okay? And then we study the concentration. So you can see that also we need to study the concentration of the chemicals involved. All right, so these are all concentration studies, but it comes to the difficult one when we start to test with the sputum samples. So remember, the blue one means positive TB, the red one is negative TB. So we need to make sure that one is the replicates, the repeatability, and also we need to compare with the standard technique. In this case, we are comparing with microscopy. So imagine that you need to have a trained eye with, if you're using microscopy, they are searching for the mycobacterium morphological. I don't know how they search and I don't know what they search. But I do know a bit, but I'm not sure if I'm the one who looking at the microscopy, whether I can detect the mycobacterium or not. So this is a video showing us actually how we get the color generation. So, but the video is not working. So this is, remember about the specificities, selectivity study and specificity. Remember about the interferences now. So when it comes to studying bacteria, okay, there are other types of bacteria that is in the sample. Okay, so we have to test whether our sensors, in this case, this is the sensors, the plasmonic ELISA, okay, are able to differentiate. Okay, this is only for tuberculosis mycobacterium. But what about other tuberculosis? And what about other bacteria that exist? Okay, so if we are able to differentiate that, then we are good to go. If we are not able to differentiate that, then our sensors are not good enough to be to work further. Okay, so those are some important characterization, yeah, typical characterization for sensors. So I'm showing you only some simple parts. One is electrochemical, the other one is optical based, okay? And of course, there are other types of transducers, and there are some additional characterizations. Sometimes we have to add some more, okay? But I'm just showing you the typical one. Okay, any questions? All right. Okay. If there is no questions, so I don't have any more slides to discuss. For the next one, 8 to 9. So I'm giving you the ample time, whether you want to uh, uh, do your homework, okay, or probably you need to rest. Okay, it's up to you.